Hi, everyone. My name is Chuck Cannon. I work at the Morton Arboretum, and I'm very disappointed to not be speaking to you in person today because I know it's a beautiful place where you are. But I do feel fortunate to present to you some work that we've been doing to construct the tan oak pen genome and to analyze it. This is a growing collaboration, kind of kicked off with Brock Peters at Advanced Genomics Technology Lab and Complete Genomics. But we've been growing this, taking in more collaborators with Matteo Garbaletto, playing a big part as well. And I'll be going through the slides very quickly. So if you want to look at something's interesting to you, this is going to be available on the YouTube channel, and you can stop the slide and look at it and send me your questions later on. But I'm going to be moving very quickly because this is a lightning talk. So anyway, so main two questions are comparative genomics. Does being a relictual species in a species poor lineage affect genome evolution? And also resistance to sudden oak death. Can the genetic basis of resistance to SOD be discovered and characterized in the pan genome? So the first question is related to the idea that the tan oak lineage is relatively unique in the Fagaceae family or the beech family because it's one of a few genera that have few species in them, but they're quite old. So it's an ancient relictual genus in North America that has not really ever diversified. So how would this affect the genomic evolution of the lineage? And the questions are, is overall genomic diversity different in species from species poor, narrowly endemic clades like tan oak compared to those in species rich, broadly distributed clades like oaks? Another question is, could integressive hybridization play a role in the diversification and maintenance of resistance and immune response genes, like those related to SOD and possibly chestnut blight? So the idea is that possibly hybridization in species-rich lineages kind of allows an accumulation and enrichment of those resistance genes, and that's perhaps why things like tan oak are more susceptible to these kind of invasive diseases than the species-rich lineages like oaks. So in the other part of the question is sudden oak death. It's a tree disease that's caused by Phytophthora. And so to address this, we collaborated with Matteo Garbaletto. He's got a trial going on where he's got 12 mother trees that have a range of resistance and susceptibility to SOD. And so we've sequenced these along with one wild tree. And you can see there is some separation in a general PCA analysis of these different groups, susceptible to versus resistance. 3 stages of assembly. First, a de novo assembly of the best genome, stitching together with the other genomes, and then gap filling with nanopore sequencing. And so we have a final draft reference genome that you can see is quite high quality, very contiguous and big assemblies. And so this has been put together by the team led by Brock Peters, and looking at the Kmer genome size and heterogeneity, you can see that there's not an obvious lack of heterogeneity in the tan oak genome there in the red box. So, you know, this will require some further analysis and thinking about what's going on there. In terms of those genes that seem to be related to immune response, like the NLR genes. So you can see, if you look at the Fagaceae taxa in this table, both chestnut and tan oak seem to have substantially fewer genes than oak and European beech. And so in looking at some Go terms of the polysaccharide binding and electron transfer activity genes, these are enriched in high D and DS genes. And there are evidence of positive selection among some of them. And we can see that those have high homology to defense response genes. And so of the 10 genes in that polysaccharide binding category, 6 have homology with leaf rust resistance 10 receptor kinase-like genes, and 2 have homology to other cell wall kinases. So that's a very quick view of the first assembly of the tan oak pen genome. We hopefully will be following up with further analyses and publication. Thank you very much.